Okay, let's get started. Welcome everybody to this educational session on host genetics and COVID-19. My name is Joas Veld and I will be chairing this session. And of course, this is a session that we are very interested in because we've all come to realize how important it is to understand more about host-virus interactions during the pandemic, but also very much about how we can detect viruses rapidly and accurately. We have two amazing speakers today. One will present online and one will be in person. We start with Dr. Kenneth Bailey. Kenneth is a clinician from Edinburgh, specialized in anesthesia, worked as a visiting scientist at Harvard and has worked with the WHO on H1N1 influenza, MERS and Ebola. He has led, importantly, several UK-wide studies during the COVID outbreak. He leads a research group at the Roslin Institute at Edinburgh and works as a consultant in intensive care at the Royal Infirmary in Edinburgh. Unfortunately, he was required to help with the UK response to the unexplained hepatitis outbreaks in children that are happening at the moment, so he will therefore give his talk online, but he is with us to answer questions afterwards. So the floor is to you, Dr. Bailey.
Well, good morning. I'm delighted to join you in Vienna. I'm very sorry that I'm not personally with you for this session. I was planning to come, but unfortunately the outbreak of hepatitis and monkeypox happening at the same time has meant that I have to stay here in the UK. I'm going to tell you about some of the work we've been doing in my lab over the last 10 years about host susceptibility to viral infections. But for obvious reasons, we're going to focus on COVID-19. At the beginning of the outbreak, we were faced with this very fundamental question. And one of the first pieces of evidence that helped us to understand this came from autopsy studies. These data come from a study led by David Dorward here at the University of Edinburgh and show slices through human lung from patients who died, or from one patient who died from COVID-19. The image on the left shows a standard stain with a slice through a pulmonary arteriole. You can see the muscular wall of the arteriole there and lots of inflammatory cells in the tissue around about it. And importantly, David stained these tissue slices. This is a longitudinal section through a similar pulmonary arteriole, stained for a variety of immune cell markers. And this marker, MRP8, indicates myeloid cells, probably monocytes, infiltrating into the wall of the blood vessel. So this is one of the first indications that a host-directed inflammatory process driven by myeloid cells was driving disease in COVID-19. But David also saw something very interesting, which is that these inflammatory infiltrates were very restricted to the lung. If you looked at ileum, that's a section of gut, this is stained for SARS-CoV-2. The brown stain indicates presence of viral proteins, indicating that the virus was there and replicating in these cells. But there's no inflammation at all. The same is true in liver. The brown stain indicates the presence of SARS-CoV-2, but no inflammation. And in fact, adding up the data from multiple autopsies, you can see highlighted in green here are all the organs in which SARS-CoV-2 was found, including skeletal muscle, but the only places where inflammation or abnormal histological findings were found were in the lungs and the spleen, where there was a reactive picture suggesting antibody production. In fact, even patients who died in intensive care, these ones highlighted here, after 25 days of infection, often had COVID-19 protein detectable in skeletal muscle and a range of other organs, but the only inflammation was present in the lungs. So that indicates an interesting and unusual tissue specificity for the inflammatory consequences of the disease. The best evidence we had suggested that the immune system, the host immune system, was driving severe disease. So the next question is, what are the mediators that are driving that? And here we were very fortunate because in the UK we had set up a research protocol. It's called the E08 Clinical Characterisation Protocol. It's already established in many countries before the outbreak, and it was the product of a consensus process among experts from a broad variety of domains across the whole world that I led back in 2012. In fact, the protocol that we came up with was used for the very first description of COVID in Wuhan at the beginning of 2020. In the UK, we had it established in every hospital here, with a system established where samples would be sent to high containment labs in Liverpool and Glasgow, and a data analysis platform that we rapidly established here in Edinburgh. And we looked initially at the cytokine expression profiles across the population and saw quite interesting patterns. So as you can see here, we clustered cytokines according to their expression across the population of patients who were hospitalised. And this group of cytokines are particularly highly expressed in patients with severe disease. And this includes cytokines including IL-6, now known to be an effective therapeutic target, and a variety of other pro-inflammatory cytokines, including GM-CSF, which, as you can see, we highlighted in the title of the paper as a potential driver of this network of pro-inflammatory cytokines. And we showed that GM-CSF is elevated throughout disease significantly in patients with severe disease compared to those with moderate disease who are hospitalised. But of course, that signal is only an observational association. It doesn't allow us to infer causality. To do that, we need either to do an interventional trial or to use genetics. And that brings me to the main purpose of my talk today. The beauty of genetics is that it does allow us to draw a causal inference, albeit with some limitations, about the potential effect of intervening at a particular genomic site or even a mediator. That brings me to the genomic study, which is a global host genetic study of susceptibility to critical illness that I lead from the University of Edinburgh. We have focused on the critically ill population with a variety of phenotypes in the target range, although the very first inclusion criterion when we wrote the protocol back in 2014, the very first thing that we included was critical illness due to emerging infections such as SARS. So genomic was a study of COVID before COVID ever existed. And I think the phenotype that we've chosen, the narrow clinical description of the patients, is the main reason why genomic is the largest source of information about the host genetics of COVID. And I think that's best exemplified by this data. This is data from the recovery trial showing the effect of dexamethasone. So as everyone I think in the audience will know, dexamethasone is an effective treatment for COVID if you have severe disease. But that's really the subgroup analysis, pre-specified subgroups in the recovery trial that revealed that. So you can see the summary of all groups together shows that treatment with dexamethasone among hospitalised patients with COVID is on balance better. The 28-day mortality is lower among this group, but there's a clear and statistically significant difference between patients who were randomised when they were already on a ventilator, just requiring oxygen, or hospitalised patients who did not require oxygen. And I think from their lack of a need for oxygen, we can infer that they didn't have severe inflammation in their lungs. The group of patients we recruited into genomic is this group here, or at least it's most similar to this group here. The vast majority of the patients that we recruited were on invasive ventilation at the time of recruitment, and those who weren't were deemed to be at risk of needing invasive ventilation. That's why they were in critical care. And that's a very narrow clinical phenotype. These patients look similar to one another. They have a slowly progressive inflammatory mediated lung disease, and so it's not just an extreme phenotype, but also a narrower one. The patients who are admitted to hospital but don't require oxygen are more clinically heterogeneous. And I think that's one of the main reasons for the success of the genomic study. The other, of course, is that we've recruited very large numbers of these narrowly defined patients. At the present time, we've got 22,000 patients recruited to genomic. And I think they're the main reasons for the success of the study. Genomic is a global study. It always has been. Back in 2015, we opened it to recruitment anywhere in the world, and we made our protocols and supporting documentation open source so that anyone can download and use our source code, if you like, for the study. And we're able to quantify how effective our design has been by comparing to other studies. This graph shows a comparison across a meta-analysis of studies from our collaborators in the SCOURGE consortium, in 23andMe and UK Biobank, and in a range of other studies that were brought together by the Human Genetics Initiative led by Andrea Ganna and Mark Daly. This shows the contribution to the signal in one gene, OAS1, from each of the contributing studies, and genomic is this tall bar in the middle, contributing about half of the signal. This is for the severe illness phenotype, which was very kindly transposed from the genomic protocol into the Host Genetics Initiative study. But the pattern is the same for the hospitalised phenotype, and it's a similar pattern across a range of different variants that have been discovered by genomic and by other studies. Genomic is the source of around half of the information, and by far the most of any of the individual studies combined in our most recent meta-analysis. Our study reported its first results in 2020, 5 months after the first critically ill COVID patient arrived. We discovered 4 new genetic associations and replicated one. This was discovered by an incredibly fast GWAS that reported about a month and a half before our study from a group of scientists in Spain and Italy. This Manhattan plot shows our unfiltered results. This was a slightly messy comparison. We compared cases in the genomic study against controls from UK Biobank, but they were genotyped on different arrays. And so there was a fairly high risk of false positive associations. We filtered our results by comparing to 2 additional control groups, one from another population genetic study called Generation Scotland, and one from the 100,000 Genome Study. And together, those studies enabled us to filter down to 5 associations, including these 4 which replicated OAS1, DPP9, TYK2, and IFNAR2. We've now gone on in partnership with Genomics England to whole genome sequence 7,400 of the cases that we recruited into the genomic study and compare them to controls from the 100,000 Genome Study. Again, population controls, and this reveals 23 genome-wide significant replicated genetic associations with COVID. I'm not going to go into the details of all of these, but I'll highlight a couple that have particularly easily comprehensible mechanisms. One is this gene IFNAR10, interferon alpha 10. It's shown in cyan in the middle here in complex with its receptor, which is composed of 2 subunits, IFNAR1 and IFNAR2, and IFNAR2 was actually an association in our first genetic study. As you can see, there is a group of amino acids in the center of the interferon alpha 10 molecule. These 3 amino acids form an interaction that holds, or is thought to hold, the protein together. And one of these is substituted in the lead SNP associated with COVID, substituted for a smaller amino acid that is predicted to prevent protein folding and hence decrease signaling through the interferon alpha pathway, at least for this one subunit. And it's that association, it's that decreased signaling, abnormal folding variant that's associated with severe disease. So I think that gives us a fairly clear direction of effect. Similarly, another association in the protein shown in orange here, PLSCR1, I think has a similarly compelling direction of effect. It's shown here in complex with a protein called importin, which imports it into the nucleus. PLSCR1 carries an inflammatory signal into the nucleus, and it's dependent on an interaction between 6 amino acids, of which one is shown here, and importin. And it's known that disrupting that signal, it's called a non-canonical nuclear localization signal, prevents PLSCR1 from being transported into the nucleus and prevents it from carrying that inflammatory signal. So again, I think a fairly clear direction of effect. Less signalling through that pathway is associated with more severe disease, and PLSCR1 is also stimulated by interferon. Combining the results now from the microarray genotyping in 22,000 patients in genomic and in meta-analysis with all of the other studies that have already been published, we now see 41 genome-wide significant associations with low heterogeneity, which we haven't published yet, but the data are freely available. Anyone who wants to download the data from this analysis just needs to go to our website shown at the bottom here, genomic.org. That's genomic with 2 C's. And you can access the data. We hope to have it published soon, and we're already seeing a range of interesting biological signals that I'll try to synthesize for you at the end of my talk. One of them is in a gene called MUC1, which we've used summary Mendelian randomization to show that expression of this gene correlates well with the impact of a given allele on the disease phenotype, critical illness with COVID. MUC1 is a mucin. It's the most abundant mucin in the lung. It's produced by airway epithelial cells, and has not just the effect of aggregating and assisting with clearance of airway material, but also has some inflammatory and immune effects. It is a therapeutic target. There are drugs that target this system, and again, perhaps less directly, but we have evidence of a direction of effect from this Mendelian randomization result that suggests that decreasing MUC1 would decrease the incidence of critical illness. So that's potentially a therapeutic target, but I think needs a bit more work. As I mentioned in our first study in December, published in Nature in December 2020, but it was we released it online at the end of August 2020. We highlighted one gene, again from a summary Mendelian randomization approach, that suggested a specific therapy, baricitinib, would be likely to be effective. Now, there's a bit of nuance around the interpretation. There was a Mendelian randomization association suggesting that more type 2 would be associated with more severe disease, but in linkage disequilibrium with a protein coding variant that is predicted to have the opposite effect. At the time, we had 9 drugs that were in the frame for inclusion in the recovery trial, and very little evidence on which to base those decisions about which drugs to include next in clinical trials. We were essentially clutching at straws for evidence on which to base the decision about which drugs we should try next. This genetic evidence pushed us in the direction a little bit more of studying baricitinib. And in addition to some clinical evidence, there were small clinical trials already suggesting benefit. But it was the genetic evidence that put baricitinib to the top of the list. So we included it in the recovery trial. And I think, again, in part because we had this genetic evidence, we had the confidence to include it in addition to the other 2 treatments that were known by this point to be effective in COVID, both of them also discovered by the recovery trial, dexamethasone and tocilizumab. And we began randomizing patients shortly after making this discovery, and as I think many people will now know, we discovered a few months ago that baricitinib is an effective treatment for COVID, and the effect is not different if patients are already on tocilizumab and steroids. So this is a third additional immunosuppressive treatment that decreases mortality in COVID. I'm very proud to say that the recovery trial led by Martin Landry and Peter Horby at the University of Oxford is responsible for several discoveries of effective treatments in COVID that together have decreased the chance of dying if you're critically ill with COVID by more than half. And I think we can start to put that evidence together from both clinical trials and from our genetic study into a model of what might be happening in patients with COVID. So in this diagram, I've assigned all processes initiated either by the virus or by the host immune system into two categories, either bad or good. And what I mean by that is perhaps whether they increase or decrease your chance of being alive 1 month after you've been infected. Of course, the processes initiated by the virus are probably, broadly speaking, bad for you, and the processes initiated by the host immune system, for example, the production of antibodies against the virus, are probably good. But I think we have to keep an open mind and be open to the possibility that some of the processes initiated by either the virus or the host have the opposite effect. In patients who fail to mount an effective immune response during the first phase of infection, it's likely that they either have higher viral loads or have prolonged viral replication. There are several genetic associations that we've discovered that are consistent with that. I mentioned PLSCR1, interferon alpha 10, and the interferon alpha beta subunit 2, receptor subunit 2. Each of these is predicted to decrease the host interferon response to viruses and increase your chance of becoming critically ill. So perhaps failing to control the virus in this early phase of disease enables the unrestricted viral replication and then stimulation of harmful inflammation. But it is fairly clear, I think, from both host genetic and therapeutic evidence that there is a second phase of the illness in which host-directed inflammation is the predominant driving factor in disease. And of course, the best evidence for that, I think, comes from the clinical trial results. So we know that this is a steroid-responsive inflammation. We know that it is inhibited by tocilizumab, and now, in part because of the genetic evidence that we've discussed, baricitinib. So these three anti-inflammatory therapies together decrease your chance of dying with COVID in this phase. But we also know that steroids have the opposite effect in a phase of the illness when hypoxic lung disease was not present. And so possibly we can assign some of the genes that we've identified that are key mediators of host inflammation, particularly myeloid inflammatory processes, particularly in the lung, that may well be driving disease in this latter phase of illness. And I've included in this factor VIII and another factor that's involved in blood clotting, which is another feature of this later phase of illness, particularly blood clotting in the lung blood vessels, which promotes a shortage of oxygen getting into the blood. I'd like to finish, before I tell you about our plans for the future, by reflecting that in COVID we now have 3 effective treatments that are host-directed. They dampen the immune response and they reduce mortality in critical COVID-19, and 41 robust host genetic associations with critical illness. In other diseases that are equally devastating, sepsis, for example, we have none of either. Sepsis kills between 10 and 20 million people around the world each year, and it's remarkable that we have made so little progress in treating sepsis, despite 40 years of intense effort to do so. And I think our experience with COVID-19 should give us hope that taking the same approaches in sepsis and other forms of critical illness will yield more progress in the years to come. And I'd like to close by telling you how I intend to do that in a programme of work that we're now well supported to deliver here at the University of Edinburgh. I've talked about the global genetic study, genomics. That's obtaining genetic signals that I hope will point us towards therapeutic targets in other forms of critical illness, as they have in COVID. We'll then be able to identify subgroups of patients who share mechanistic biology, which is an unmet need, particularly in critical care medicine. The need is not just for effective therapeutic targets, but to identify which patients those therapeutic targets are likely to be effective in. And genetics, I think, has a great deal to offer here, in large part because of the colossal amount of spare statistical power that there is in genetic studies to enable us to do retrospective subgroup analyses and identify differential genetic associations in different clinically determined subgroups of patients. So together with identifying the targets and the patient populations, we'll be able to design clinical trials in silico using genetics. And then if we're lucky, and I think it's fair to say we got very lucky indeed with the type 2 association that predicted parasitinib, then it might be that we can do that again and go straight into clinical trials. I'm not aware of that having happened before in infectious disease or critical care medicine. I think the baricitinib type 2 association is the first time we've gone straight from a genetic hit into an effective treatment, but it's entirely possible that we'll be able to do it again. I think it's more likely that we'll have to do a bit more work to identify effective treatments based on the in silico trial design. And so working with my colleague, Professor Kev Dhaliwal here at the University of Edinburgh, we're developing technology to use a robot bronchoscope to deliver tiny doses of drugs into small regions of the lung in critically ill patients with the actual disease that we're trying to treat, in order to determine whether those drugs are likely to be efficacious and to prioritise effective treatments for inclusion in large-scale clinical trials. There's a concept that we call micro-trials. Much of the technology already exists, and we anticipate that we'll be able to do this within 5 years. I'm going to stop there and acknowledge my collaborators. I've listed the co-authors on our paper here, but I think the most important people to thank are firstly the research nurses who, at considerable personal risk and in really circumstances that were really unimaginable at the start of the outbreak, recruited thousands of patients into the genomic study. There are 1,430 research nurses listed on this slide, and every single one of them contributed to making the genomic study a success. This is what's really needed to get genetic research to work. And of course, the patients themselves, who at a time in their lives when their own life was immediately at risk, generously agreed to contribute to a research study at no potential benefit to themselves, just to help other people. I will stop there. Of course, I have to acknowledge our funders, our partners, Genomics England, who did the whole genome sequencing for the middle one of the 3 analyses that I showed, and my friends and collaborators across the UK. Thanks very much indeed.
Thank you very much, Dr. Bailey. It is a fascinating story. It shows you how important genetics is in critical care, but also shows how much we have to do, which of course is very challenging, but also, of course, says something for our field. There is already lots of people lining up, so it's good that you kept your talk relatively short, so we have lots of time for questions. I'll start on the right for me. So yes, introduce yourself, please, and ask your questions.
Sven Bergman from the University of Lausanne. Thank you so much for this fascinating talk. I want to ask about the last point you raised, where therapies can be more stratified and informed by the genotypic information. Now, the genetic architecture of this trait, do you think that it really will be not too polygenic to make real groups? Because ultimately it's random and there's no guarantee that not too many people will fall in between many different risk factors and not fall into groups which will be profit from targeted specific drugs.
Thank you for your question. I'm very grateful for you raising that point. I was meaning something slightly different.
So the problem we have in diseases, particularly like sepsis, is that the doctors who are treating the patients don't understand the pathophysiology. And we have good reason to believe that patients with sepsis and other forms of critical illness fall into groups that are biologically different. They have different diseases, and we've grouped them all together in this one rather incoherent syndrome. The way I think genetics can help us there is that we can test for biological coherence among hypothesized subgroups. So we can take a group of patients who have a particular pattern of clinical features or immune response markers who might be a distinct disease, and where in the past we would have been, in order to test whether they were really different, we would have had to do a clinical trial. We can use the possibility that there will be differential genetic effects between that subgroup of patients and another subgroup to infer that there's a really biologically meaningful difference in the disease mechanism. I think one reason I have hope that will be effective is that if you take the p-value from the recovery trial for the effective interventions that we've found, the p-values range, they're sort of in the range 0.001 to a little bit higher. The smallest p-value in our latest analysis of the genomic study is 10 to the power of minus 249. So there is a colossal amount of spare statistical power. That means if you divide the population in a clinical trial into subgroups, you immediately run out of power. You can't distinguish between them, whereas because the genetic signals, or at least the strongest genetic signals, are so much more robustly detected, we have the potential to separate subgroups and then know that they're really different.
Okay, thank you. Thank you. We go to the middle. Yes.
Thank you very much for this very interesting talk. My name is Max Witzel from Munich Medical Genetics Center. My question is there already any point in for personalized medicine or for testing patients on this 41 loci you determined, or do we need to wait for a kind of PRS score which balances the advantage and the disadvantage of genetic traits on COVID?
So two elements to that question. I think the possibility of personalised medicine in COVID-19, I think is not an immediate opportunity. We need, in pandemics, when we're suddenly hit with a tidal wave of patients with a new disease, we need effective, ideally cheap interventions that can be delivered at very broad scale worldwide. So we were incredibly lucky that dexamethasone was effective, and we found it in the recovery trial and was cheap and deployable across the whole world. Anything that requires a test to be done before treatment is obviously going to be much more complex to deliver in clinical practice, more expensive, and more difficult to deliver to the world's population. So that has never been an urgent priority for the COVID-19 work. We will do a polygenic risk score we're working on it now in our current analysis of the genomic study. I think we have enough data now to calculate a polygenic risk score, albeit for obviously a predominantly UK population. And I think that will be interesting and important, but I don't think it's going to immediately change the way we treat patients. There's a potential, the utility of it might be things like if we have a population that is well genotyped or can be genotyped quite easily, we will be able to target therapies that are not widely available. So antiviral therapies, monoclonal antibodies that are effective in that early phase of disease when there's lots of virus around, but are not widely available. We have to target them as best we can in the population to patients who really are at very high risk of death or critical illness from COVID. And it's possible that a polygenic risk score might have an impact there. right now, in some populations that are already well genotyped, and realistically more in the next 5 or 10 years when genotyping is more broadly available.
So there's no point in testing patients for your 41 genome loci you identified associated with severe COVID?
I don't test my patients for those variants now. I think by the time a patient meets me, we already know that they're desperately sick and they're susceptible to COVID. The urgency behind this work is using genetics to find therapeutic targets, to find ways to make everyone better, rather than testing people for genetic markers that would identify their potential to benefit from a specific treatment. In future, it is realistic, I think, to think that we might well be able to use genetic risk prediction to stratify therapy, where therapy is limited or costly, either financially or in terms of risk to the patient. But right now, in the real world, that isn't something that we can deploy at large scale.
Okay.
Thank you.
Let's go on and try to ask short questions and quick answers. So we have, because there's lots of people behind the microphone. Yes.
Thank you for this beautiful talk. It's Mario Capasso from University of Naples. The question is, are all your patients infected with the same strain virus? And if not, are you consider this bias, potential bias?
They are not all infected with the same strain of the virus. We do now have whole viral sequence data for 1.3 million people in the UK in the same computer. And it's actually in the last 2 weeks that it landed in the same computer in our outbreak data analysis platform here in Edinburgh. So we'll be able to do a combinatorial host viral genetic study. Of course, we'll have much less statistical power to detect differences between strains of the virus. There are interesting signs, though, and I will be brief, but many people have noticed that in our first study, we reported a gene called E08.1, which is a host antiviral gene, was associated with susceptibility to COVID. That spawned a whole lot of work in virology. In our second analysis, it was no longer genome-wide significant. And I think that's because of a change in the virus rather than a change in the human population. But we haven't proven that yet, so sadly, I don't have results to present to you just now.
Okay, thank you.
Very interesting. Yes, question in the back.
Yeah, thank you very much. So Carsten Bachmann from Mainz in Germany. So due to the overlapping character of some complement-driven disease phenotypes, I'm actually interested to hear your opinion if you see a role for the complement system, and if yes, which role do you think it plays in the disease process?
So we haven't seen a lot of complement genes in our unbiased analysis. That doesn't mean that there aren't important effects there. I think there's clearly crosstalk between complement, myeloid immune signaling, and coagulation, and there's much left to learn about this disease. I suppose that in order to test those sorts of hypotheses, we're going to need a model system, which we haven't yet really developed a good model system of COVID. It's quite instructive how little we learned from animal models during this outbreak. I think that was partly because of the speed with which we needed all the information. There simply wasn't time. But I hope in the next few years we develop systems that will enable us to really tackle those hypotheses.
All right, thank you. Now, of course, I try to hurry on, and now there's nobody behind the microphones, but I have a question for you, Kenneth. I was wondering, in the same hall here yesterday, we had a discussion on genomics in Africa, and I learned, and many of us perhaps know, but the number was shocking, that 0.1% of participants in GWAS studies are from African descent. And of course, that has a big impact on genomic medicine. And you already said that, of course, your work is in the UK population. So, what are we missing when we think about host studies? How are you working with colleagues around the globe to improve this work and make it relevant for all populations?
Well, that's a great question. I'm really grateful you asked it. My map had Africa as a massive untouched area. The genomic study is not currently recruiting in Africa. And I've only highlighted on that map the centres where we're actually actively setting up the study, but we are very actively pursuing collaborations in both Nigeria and Sierra Leone to recruit patients there. That's through a critical care research network run by Rashan Hanifa and Abby Bean, which covers 16 countries around the world, all low and middle income countries, and is a really extraordinary collaboration that provides data collection from critically ill people in an electronic format in very low resource settings. But the quality of the data collection is actually better than in many high income settings. And it provides a platform not just for genomic, but also for clinical trials. And REMAP-CAP, which some people might recognize as one of the COVID trials, is active already in that network. So I think in 16 countries, including Nigeria, Sierra Leone, and several others in Africa, we will be able to establish genomic in the next few years and recruit not only a more diverse human population, but a more diverse clinical population, because there are diseases where there might be really important therapeutically relevant genetic discoveries to make, such as dengue, that we couldn't make if we only recruit in the places where I work. Yep. I think that's not only a scientific imperative to scan the full diversity of the human population in order to identify fine mapping signals that might lead us to lead variants that might have much more compelling causal relevance, but also, of course, a moral imperative, as you suggest.
Yeah, no, absolutely fantastic. Perhaps then, for a last question, what would you say to all these young colleagues here in the room, genomicists and computational scientists who think about a career in this field? This is something that perhaps not all of us in human genetics thought about, but we now see, a lot of need for this.
A need for young people to study genetics?
No, well, in critical care, in infectious disease,
yeah.
This is, in your field.
So there are very few people doing genetics and critical care medicine, and the stated aim from the beginning of the genomic study in 2015 was to build capacity around the world to answer these questions. This is a new tool. Genomics is relatively new in the history of human medicine, and clearly we've now shown has the potential to have a transformative impact on critical care. So the opportunity is huge. It's not going to go away. And so we need to train people to understand critical care medicine if they're geneticists, and to understand genetics if they're critical care doctors, so that they can lead their own studies. And the intention is that my new collaborators in Nigeria, Sierra Leone, Bangladesh, and Pakistan will lead their own genetic studies without me, and deliver their own genetic research to tackle problems that matter to them. So the main message that I have for young people or trainees or postdocs in your audience is come and work with us. Either come and join our growing and very dynamic team based here in Edinburgh, or join the project near wherever you are, because there are opportunities opening up all over the world.
Yeah, thank you very much again. Thank you, Dr. Bailey.